All right. Hello, everyone. Welcome to this new edition of the Worldwide NeuroDev Forum. Today's guest is Laurent and Nguyen from Liège University. Glad to see there's so many of you out there. Special guests from Japan, I see. So a special hi to you guys out there in Japan. I know it's quite late. Thanks for joining in live. For my part, I'm in my natural habitat back in the lab. I hope it's the case for many of you. And if it's not the case, I hope it's going to be the case soon and that things will progressively get back to normal. So there's something special actually today. We're going to be live also from Twitter. If somehow I manage to press the right buttons right now. So if you don't have access, or if you are on a, here you go. Yeah, it should work. So there's a live feed from Twitter now. Well, maybe I should put the volume away. Voila. So anyway, from now on, I think most of these worldwide neuro events are going to be broadcast on Twitter, so it makes it a little bit easier to join in. So back to today's guest, Laurent and Nguyen at the Liège University. He's a permanent senior researcher of the Belgium National Fund for Research, and he's the director of the GIGA Stem Cell Institute at this university. So Laurent did his PhD in Liège under the direction of Gustav Nguyen from 1999 to 2003, and received an EMBO long-term fellowship to do his postdoc in François Guillemot's lab in London. So that was from 2003 to 2006, so a relatively rapid early career, which gave him the opportunity to set up his lab at a relatively young age. So Laurent is studying the cellular and molecular mechanisms that control the development of the cerebral cortex, with a particular interest in neurogenesis and neuronal migration in health and disease. His research has been rewarded with quite a few prizes, the Simon and Pierre Clerdon Prize, the Scientific Route Foundation of AstraZeneca, amongst others. So he's published quite a large number of publications, including in some, I'm reading from his bio there, including in some prestigious scientific journals. But you know, Laurent, that it doesn't matter if it's a prestigious journal or not. What counts is if it's the type of research that you're doing is prestigious, and that's the case. In your case, I think people really appreciate the fact that you have this broad knowledge on the molecular and cellular basis of corticogenesis, and that really gives you the ability to jump successfully from topic to topic and to integrate these different molecular and cellular aspects. As always, I tried to give a little personal note on our speaker. For Laurent, I didn't have to ask him about any anecdotes because we know each other for quite a while. I actually had to strike quite a few of them out. But maybe one thing that most of you might know about Laurent is that he's a big sports fan, a big runner, amongst other things. And the other thing also is that he has a big heart. And I remember one time, Laurent, when I don't know, we were chatting you were actually doing something for a charity where you had to run indoors or do some kind of high intensity sports, and where whoever would have the highest heartbeat would win. So it sounded a little bit dangerous to me, you know, keep your heartbeat as high as possible for as long as possible. I don't know if it was a kind of a stroke prevention charity or anything, but in any case, I mean, I guess the heart analogy is one that goes well with you, because you do a lot of things with your heart, including with regard to your science. So I really look forward to listening to your work.
Thanks, Denis. Such a kind introduction. This is a great pleasure to be part of this fantastic online seminar series. It's really an honor. Thank you very much for inviting me. Yeah, I'm not unfortunately in the lab. I'm still kind of stuck in the house, but I should return to the lab soon, like most of us, hopefully. Yeah, so my laboratory, like Denis said, is really interested by the cellular and molecular mechanisms that drive the development of the cortex. We have different topics in the lab, ranging from neurogenesis to cell migration, and more recently, we've been interested by axonal transport as well. And of course, today what I want to put is some emphasis on the control of cell migration in cortical development. So I will share my screen. Okay, I hope you can see it.
Yep.
Okay, that's great. So today what I want to say, I want to present, sorry, two main stories. One is published, and we have some follow-ups.
Laurent, can you exit the full screen mode? I think you shared your screen. You should share the whole screen. You just shared the PowerPoint.
All right, sorry. Let me try again.
So close the screen sharing.
Right, and then
share the whole screen.
Oh, that was a mistake. Sorry. Oh, yes.
Close other windows you don't want us to see. All right, that's good.
Okay, so then it should work, right? So yeah, again, yeah, good. So what I want to do today is really present two stories. One is partly published, and the other one is brand new, and this is not published. I want to present you some data about the function of cell migration in the building of the cortex. So we are located in Belgium, in Liège. We are part of this institute, which is called GIGA, for Interdisciplinary Group of Applied Geno-Proteomics. And we are hosted by the University Hospital, which is actually a great place because, as you can see here, it's surrounded by woods. And as you heard, I like running, so I usually break during lunchtime for a little run with friends. Anyway, let's kick off with basics about cortex development. So if you perform a section through the head of an adult, you have this section through the brain, and the cortex correspond to the outermost and top layer of the brain. In human, it's about 2 to 4 millimeters thick, depending where you're looking at, and it's a layered structure. So it's composed by layers of cells, 6 layers in the neocortex, mostly neurons intermingled with glial cells. Now, if you look at different cortical regions, the organization of those layers is a little bit different. Like I said, most of the cells that compose the cortex are actually neurons. And we can roughly subdivide these neurons into major classes. Most of the neurons are actually the excitatory neurons that use glutamate as a main neurotransmitter. They are so-called projection neurons because they are sending long axons toward distant cortical and subcortical targets. And the remaining neurons are the GABAergic inhibitory interneurons. And on this slide, you can already appreciate the huge morphological diversity that characterize different subtypes of inhibitory or excitatory neurons. The classification of these cells actually relies not only on the morphology, but also on the physiology. And recently, with the blooming of single-cell biology, single-cell sequencing, tens of different subtypes of interneurons, lots of subtypes of projection neurons have been discovered in the mouse as well as in the primate cortex. And this is a lot of work coming from the Allen Institute. And there's also this very interesting fact that has been reported, which is if you look in different cortical region of the brain, you see that the diversity of GABAergic interneurons is quite well represented throughout the different cortical region. And I'm actually talking here about mostly the motor and visual area of the cortex. But for the glutamatergic neurons, it's a little bit different. You can find some very specific subclasses of projection neurons that are mostly enriched in some region and not others. So how do you generate such diversity? Well, it's still quite unknown, but there's information coming from single-cell sequencing that are arriving. And I'd just like to highlight briefly a very nice collaboration that we had with Denis' lab. We were actually sharing a PhD student, a bright one, Glycine, and Glycine teamed with Ludovic Tellet in Denis' lab to show that there's actually a temporal progression of the apical progenitors that can be seen as the stem cell of the cortex at the bottom of the tree. Indeed, while the recipe to make neurons is conserved throughout corticogenesis, there's actually some kind of priming of specificity which arise in the apical progenitors, and at the onset of corticogenesis, these cells can be seen as more introverted. They express a lot of genes that control the cell cycle, chromatin organization, and as time is passing, these cells transformed and start to express receptors for cues. They are starting to be more extroverted, more sensitive to cues from the environment that actually also dynamically changing. And this is how the diversity of projection neuron is imprinted within the progenitors. So if you look at the basic steps of cortical development, it all starts with the generation of neurons from those progenitors, the apical progenitors that are also termed radial glial cells. These guys divide asymmetrically to self-renew, but also give birth to a projection neurons through what is called direct neurogenesis. And as corticogenesis advances, there's actually a production of intermediate progenitors that are not so complex in the mouse, but that get very complex in primates because you have several subtypes, and I'm not going into much detail here. But basically, by dividing asymmetrically, giving rise to those intermediate progenitors, there's actually an expansion of the number of neurons produced, because these intermediate progenitors can be seen as transient amplifying progenitors, giving rise to the bulk of the projection neurons that will sit in the cortex and mostly in the superficial layers of the cortex. And after this wave of neurogenesis, there's a wave of gliogenesis for astrocytes, oligonucleocytes, and so on and so forth. So when the neurons are just born, they will then need to reach their final position in the cortex, and this is where migration is very important. And for the projection neurons, they will use a mode of migration which is termed radial migration, spending most of the journey locomoting on the fibers of the radial glial cells to find a position in the adhoc layer. For the interneurons, the story is a bit different. So there are also different types of progenitors that are this time located ventrally in the ventral forebrain region that are called the gyneic eminences. Most of those interneurons are produced by the medial and the caudal gyneic eminences, and these neurons migrate through tangential path to integrate the developing cortical and ledge. And they will be responsible for controlling local network. To cut the long story short, they are very important to control the overall excitability of the cortex. When the cells are reaching their position, they will start growing neurites and getting differentiated morphologically, even though it's actually starting, for the axon, for instance, as the projection neuron is migrating. And these axons can be sent to different places. For superficial layer projection neurons, these are mostly cortical projection, and you have here the contralateral projection. And for the deep layer projection neurons, we have cortical-fugal projection. Some are going very far away to the spinal cord for some layer 5 neurons, for instance. And these cells will basically connect to themselves. For the interneurons, their maturation is local, and they are integrating local network. So this guy will then need to form synapses to connect themselves, and this synaptogenesis process starts just before birth and is remodeled until adolescence by pruning. And this is a rendering picture from the last paper for Frank Pohle, published last week in Neuron, just to show you the distribution of the excitatory synapse on the basal dendrite from a layer 2 projection neuron. If you are interested by looking at synapse topology, please have a look at this fantastic resource paper. We believe that it is very important to better understand the mechanisms at play during cortical development, because most of the human brain malformation or cortical malformation start from an interference with the early steps of corticogenesis. And if we look at the migration process, We know that some mutation in genes coding for protein that will associate with microtubules such as double coating or LIS1, or who are at play with the actin cytoskeleton such as filamin A, can lead to dramatic cortical malformation. And you have here lissencephaly upon mutation of LIS1, for instance, and here a nodular periventricular nodular heterotopia when filamin A is targeted. And if you interfere with the proliferation and the production of these neurons, you can basically end up with microcephaly, where you can see on this brain scan the overall architecture of the brain is quite conserved, but basically the total number of neurons that populate the cortex is actually reduced. And this can be caused by mutation that disrupt genes important for the proliferation, such as MECPH, but also, and I'd like to highlight this, toxic toxic drugs, or like alcohol or viruses, and we're taking the example of Zika, that are actually transmitted vertically during interior development and leading to cortical malformation and microcephaly in particular. And we've been working a lot on this lately, showing part of the mechanisms that are at play to explain how Zika is inducing microcephaly in the mouse. So the lab is mostly interested by the basic science behind cortex formation, but we like to have rationale from clinicians, and so we are collaborating a lot with different clinicians to get information. That was my bug, my floating bug, sorry about that. From clinicians that are actually feeding us with mutations that are associated with patient cohorts suffering from specific cortical malformation, and our job is to try to understand the function of the gene if it's unknown, and also try to decipher how variant or mutation in these genes lead to cortical malformation. But today, I'd like to spend more time on basic science, and I'd like to talk about the role of migration, of cell migration in cortical morphogenesis. So intuitively, you understand that migration is important to bring cells from the place of birth to their final destination. And this is really true for the cortex. Most of the cells are actually produced distantly from the final position. And if we look at the interneuron, for instance. But this is also a way to convey information and influence the neighboring while migrating. And I'm talking just here with two examples, the CAR-H cells that are important for the layering of the projection neurons in the cortex, and these guys are coming from far away. Yeah, this is the work of many labs, and I may miss some, and I'm sorry about that. Microglia are also important. If you are impacting the migration of microglia, you may affect the fine-tuning of positioning of interneurons in layers, and this is a beautiful work from Sonia Garel, and the famous corridor cell described about a decade ago by Guillermina, Sonia, and Oscar, by the way, which is important to open the space for thalamocortical axon to grow in the cortex. So these are some examples. Today, I want to talk about the cortical interneurons, and that's going to be the first part of the talk. For the second part, which is not published, I'm going to talk about the oligodendrocyte progenitor cells. So let's kick off with the interneurons. Well, you can see here that these cells are highly polarized. They are bipolar with the soma and nucleus here, and the leading process which gives the direction of migration. So this is a time-lapse recording performed in the lab, recorded pretty much in this region of the forebrain, just to give you a feeling of how this population is migrating. It's really dynamic. They're migrating at high speeds between 30 and 50 micrometers, which is about 4 times faster than the projection neuron that are locomoting on the fibers. You have also this clear impression of discontinuous migration, and I'll come back to that in a moment. So like I said, these interneurons are born from the ventral forebrain, from the Ganic eminences, and I'm taking the example of the MGE, which produce about 60% of these interneurons. And in the Ganic eminences, there's already some diversity of progenitors that basically prime and give rise to different subtypes of interneurons. So there are two very great papers from Oscar and Gord Fischer's lab, where by single cell biology they could identify that these progenitors that are sitting in the MGE are already primed to some extent. They already show some kind of molecular signature that will imprint the differentiation of the subtype of interneurons. But what is clear is that the program of differentiation and final identity acquisition, I should say, even positioning, is actually unfolding as these cells are migrating. And until now, it's not clear whether there's an impact or an influence of the environment for fine-tuning the choice of identity and connection. And this is something on which we are actually actively working also in the lab. So if you look in 3D by transparency, you see that these interneurons are using streams of migration path, right? And you can see that the streams are not all synchronized. They are more advanced in the anterior part of the cortex. As the cortex develops, there's also dynamic addition of new streams. How are these cells finding their way to the cortex? We know that the extrinsic cues are very important to pave the path for the migration of those interneurons, and this is the case for chemoattractant or chemorepulsive, and I'm not going into much detail. There are also systems where there are receptors that are involved in this system, ligand receptors, and this basically draw the path of migration. We believe from recent work that some intrinsic mechanisms can also drive the choice of migration, and this is a beautiful paper from Lynette Lim when she was postdoc in Oscar Marin, showing that the somatostatin neurons are actually mostly using the superficial layer. So the story is quite complex indeed. So when the cells are entering into the cortex, they will have to find their way to the different layers. And now we are entering into the intracortical dispersion process. Well, these cells will arrive and use radial and most often oblique path of migration. Eva Anton also showed that a minority of these cells are engaging in interaction with radial guide process, but it's just a minority of them. And when the cells are reaching the layer, if you now look in the plane of the layer, they will have to disperse tangentially using multimodal migration modes to, I would say, populate quite homogeneously specific region of the cortex. Again, how are these cells finding their way within the cortex? Two papers published by Eva Anton's lab and Christine Mettin's lab highlighted a function for the primary cilium as an antenna to receive signaling for finding their way, for guidance, right? And you can see the path of migration. Actually, as you know, guys, the primary cilium is the platform for sonic hedgehog signaling, and there are actually traces of sonic hedgehog in the dorsal part of the cortex, and we didn't know that before, right? But they can be sensed by those primary cilium and help the interneurons in the guidance process. I'd like also to highlight a good, excellent preprint from Isabelle Caillé in Paris. They show actually that the primary cilium is also important to sustain migration per se by activating a CMP-PKA signaling at the centrosome. So the primary cilium is not only important for guiding the cell, but also for allowing the cells to move. And this is true for the interneurons, but also for the interneurons that are migrating in the rostral migratory stream after birth. Let's go back to the cell biology because, like Denis said, I'm really in love with cell biology. So I told you that these cells are actually polarized. You have the soma here, the leading process, which is actively branching. And these cells, like I mentioned, are are migrating using a saltatory migration. And this saltatory migration is made of cycles, and this is a decomposed cycle. You have a first step of centrokinesis. So the centrosome, which is the head of the nucleus, is moving within the leading process in the bulge. This is then followed by a quick translocation of the nucleus, which is called nucleokinesis. And at the same time, you have a dynamic branching of the process where the growth cone-like structure is basically splitting in a dichotomic way to help also the neuron to find its way in the forebrain parenchyma. So what is the molecular machinery promoting the migration of these cells? And I'm now talking specifically of these cortical interneurons. Well, we know that most of the force is coming from behind. This is about actomyosin contraction, highly polarized behind the nucleus and propelling the nucleus forward, which basically pays the migration of the cells. There's a little bit also of treadmill activity from the actin retrograde flow here, but this is a minor contribution. What is different with SVZ interneurons, for instance, is that there's no tight link between the centrosome and the cage of microtubule that englobe the soma, because in the SVZ there's really a pulling force, which is actually not seen here. The reason is that, one proof is that the nucleus is often overshooting the centrosome, right? So the migration of those interneurons is complex and depends on an interplay, like I mentioned, between extracellular cues and intrinsic regulators that are ultimately converging on the dynamic remodeling of the cytoskeleton. And this is really something on which we are looking at and we are excited to better understand. So what I'd like to do now is to exemplify this kind of crosstalk between, or coordination, I should say, between extracellular cues and intrinsic regulators. So let's go back in time, 2013. Ariel Avila Macaya, who is now a PI in Chile, was PhD at that time, and he was interested by glycine as a possible ambient neurotransmitters having an impact on the migration of those interneurons. And glycine is actually the smallest amino acid neurotransmitter found in the brain, which can actually activate these ligand-gated ion channel receptors that are called glycine receptors, made of 5 subunits, alpha or beta. And if you look at the distribution of glycine during forebrain development, you see that you can detect glycine both in the ventral but also in the dorsal region. And what Ariel showed is that glycine receptors are actually nicely expressed in these migrating interneurons that are highlighted by GFP. And if you look at the distribution of the different subunits, you see that during embryogenesis, these receptors are indeed alpha-2 homomeric, which is confirmed by pharmacological analysis using patch film, where glycine elicitates inward current, and this is blocked by pitrotoxin and pitrotoxin, which really suggest and support the idea that they are alpha-2 homomeric. So what happens if these receptors are not expressed? We found that there's a strong reduction in migration, and this is coming from a reduction in the speed of migration, as found by Ariel. It was basically using a medium containing a defined concentration of glycine with or without strychnine, which is a specific blocker of the glycine receptors. And by adding glycine, he found a reduction in the migration speed. We had exactly the same data by knocking down acutely alpha-2 in these cells. Why a reduction in migration speed? Because there's a decrease in the frequency of the nucleokinesis. So this movement of the nucleus, there's less of it. So glycine receptors are very well known as inhibitory receptors in major neurons. But now we're dealing with progenitors. And in progenitors, there's an inversion of the chloride gradient. So what happens if you trigger the activation of those receptors? This will actually trigger membrane depolarization. And what we found is that this depolarization trigger the opening of voltage-gated calcium channel and allow the entry of calcium, which is linked with a burst of contraction as seen with this probe for actomyosin. In a nutshell, activating glycine receptors trigger membrane depolarization, allow the entry of calcium, and that's important for the activity of that kinase, which is called myosin light chain 2 kinase, which is responsible for the phosphorylation of the light chain of the myosin 2 complex, which is this non-muscle myosin involved in the actomyosin contraction. And the phosphorylation actually allows the myosin 2 to acquire its functional form. So it looks like, because glycine is ambient, glycine is just feeding the system, it just sustains migration, basically a bit like fueling the engine of these interneurons. And now I'd like to talk about an intrinsic regulator, which is called P27. So that's also back in 2012 or 13, I don't remember, when actually Juliette Godin, who was a postdoc at that time, she's now a very successful PI at IGBMC in Strasbourg, was teaming with Sophie. Sophie is a PI to become, right, very soon. And they were interested by P27. P27 is a protein that belongs to a family of CYP proteins, P21, 57, and 27. And they've been described in the 90s by the lab of Jim Roberts and others as being mostly involved in promotion of cell cycle exit by blocking this couple of CDKs and cyclin. But together with a postdoc in Jim Roberts' lab, Arnaud Besson, we discovered in the Cortex that P27 has also function that goes beyond cell cycle regulation. And what we tested here is the function of P27 in the interneurons. The reason is that P27, as you can see here, is beautifully enriched in these migrating interneurons that are far away from the place of birth. And P27, you can even notice that it's not only in the nucleus, but is also leaking in the cytoplasm, suggesting cell cycle independent function. And by looking at this, the mouse coming from this breeding, so the idea is that you conditionally remove P27 from newborn cortical interneurons, and you have the GFP to track the cells. We saw a massive disruption of the migration. That was actually a delay of migration due to a reduction in the speed of migration at all rostrocaudal region that we looked at. So I told you that glycine feeds the engine. It basically allows this pathway to promote the movement of cells through contraction behind the nucleus. I didn't mention it, but it's also important for the branches, right? And what Juliette and Sophie discovered is that P27, nicely colocalized with RhoA, physically bind to RhoA, blocking its activation by the GEFs, and by doing so, basically put a brake on the pathway. Now you would say, how can this work? You have, at the same time, this positive pathway and this brake. What we believe is that the brake is cycling because P27 is shuttling a lot from the cytoplasm to the nucleus and back, and it can also be degraded through different pathways. So the interplay between the cycling of P27 and the tonic activation of the pathway by glycine and likely additional cues is driving the behavior of these interneurons, this saltatory migration. Opening the bracket here, P27 is also a microtubule-associated protein that can promote the polymerization of the microtubule in the extending neurites. So it's very important for the branching, the dynamic branching. And if you're interested in, we've been also looking at P27 later on because it is also expressed in mature neurons where it actually controls the transport of specific cargoes. Now, there's a long-standing question in the field. So why are these cells undergoing such a saltatory migration? It's extensive. You can see how big is the jump from the nucleus. This migration is made of cycles of pauses and motion. And so I guess the question is, what is the function of pausing during migration? This is really a tough question. And we are lucky because we had two great postdocs in the lab. Actually, Carla is still with us, and she's looking for a PI position, so she's on the market right now. And Elise, she's now working for a private company. well, by teaming, they were able to ask this specific question. So if you look at the history of the lab on tangential migration, and I'm just showing 3 examples here, we looked at several mutants, P27, glycine receptor, I just mentioned them, elongator, we've been looking a lot at this complex. You can see that losing elongator affect the migration too. Indeed, when you lose any of these molecules, we end up with super complex migration defects, including a mixture of branching defect, nucleokinesis defect, and reduction in migration speed. And with such a complex migration defect, we cannot ask the question of the posing during migration, because you would expect to keep at least the same average speed, right? And I told you that the lab is interested by different topics. We are also very interested by axonal transport and post-translational modification taking place on the microtubules. One of them is polyglutamylation. And we were looking at that mouse, and we were lucky, actually, that this model, as you will see, can allow us to ask this specific question of the posing during migration. So, in a nutshell, polyglutamylation is a post-translational modification firstly described on alpha and beta subunits on their tails, right? And you have polyglutamylation, which is the addition of glutamate. You have this branching point glutamate dependent on the activity of those TTLL for tyrosine, tubulin tyrosine-like ligases, sorry. And then you have others that are extending the chain. And you have the converse enzyme that are digesting the chain, which are called the carboxypeptidases. And you have CCP5, which is the one removing the branching point glutamate. So there's a nice correlation between the level of glutamylation and function of microtubule in terms of transport and severing. And I'm inviting you to read, if you're interested in, this very recent paper, this review, sorry, from Carsten Jenke lab. Actually, there's a whole bunch of proteins that are glutamylated, which suggests that glutamylation made a broader impact on the proteome, right? So why have we been looking at that for the interneurons? Because we found that the interneurons were massively enriched in glutamylated protein, as simple as that. And this is a clarified brain from this mouse line. You can see basically the interneuron stream, right? You navigating within the cortex. And we use this model to purify those interneurons by FACS, and we analyzed by QRT-PCR the messenger content for the different enzymes involving glutamylation. And we were struck by the selective enrichment for some CCPs and some TTLLs, and we decided to focus our attention on the CCPs. And CCP1 in particular, reason is that this is the one important for digesting the chain, while CCP1, 5, would only work if the chain is removed to remove the branching point glutamate. So when you look at the distribution of the messengers by institutionalization, you can see on this section through the E12.5 embryo, a beautiful enrichment of those messengers in the domain where these internals are produced. You can even guess, to some extent, the initial form of migration of those cells that will at some point populate the cortex. At the cellular level, by immunostainings, you can see that CCP1 is distributed everywhere in the cytoplasm, enriched in the growth cone-like structure around the nucleus. There's also some in the nucleus, suggesting that it can actually shuttle from and out the nucleus. So what we decided to do was to try to test the function of CCP1 in interneurons and test what would be the impact of keeping long chain of glutamate on protein in these interneurons. For this, we used this breeding. We had a chance to collaborate with Carsten Jenke and Annie Andrieux for the mouse. And we did some time-lapse recordings. And we combined 2 different approaches. The first is an approach that was initially described 15 years ago by Christine Mettin. And I must say that I'm really happy to have collaborated with Christine. She's a fantastic scientist. And I really learned the basics when I was starting my lab to track these cells and understand the behavior of these cells. Anyway, what she developed is a co-culture system where you would basically culture MGX plant on homochronic mixed cortical feeders. And then you can reassess the details of migration, such as the jump of the nucleus, the branching of the leading process. What you miss here is really the directionality. And so for the directionality, what is good is to do organotypic brain slice recordings. So then you can really see, for instance, here cells entering into the specific streams. So using those two approaches, we decided to test the impact of losing CCP1 in migrating interneurones. Just to remember, you that with this mouse line, we would remove CCP1 in post-mitotic cells, not in their progenitors. And on this graph, what you see is actually the tracking of the soma of a representative control, CCP1, or a representative CCP1 conditional knockout interneuron. And you see each bump as a nucleokinesis when it is above 5 micrometers. And first thing that we saw is that there's no changes for the average speed of migration, which is very important for later. But what we saw, and you can actually guess it from the graph, there's a reduction in the average amplitude of nucleokinesis, which come together with a reduction in the total time of nuclear posing. So if you look at the poses of the nucleus, when these dots touch this axis at 0, you see that the wild type is actually stopping more, right? And in other words, what we have are less amplitude in nucleokinesis and more movement of the nucleus. So reduced posing during migration without affecting the migration speed. In other words, we are looking here to a conversion of behavior of migration from a saltatory migration to a less saltatory migration. I would even say a sliding movement. With this model, then we can assess the function of posing during migration, since we are not affecting the average speed of migration. Yeah, we looked at the branches as well, because I told you that glutamylation is important for the microtubule biology, and one readout is the branching. What we noticed is a very specific defect in the half-life that was extended for the secondary neurite, okay? And this actually could be counteracted by, at the same time, targeting acutely TTLL1, which is responsible for the extension of the chain. While we were able to rescue the branching dynamics, the behavior of the cell as a whole wasn't changed. It was still gliding. So I told you that the mechanisms that drive the migration of these cells is mostly this actomyosin contraction that are very well polarized behind the nucleus, right? And this is a way with a live prop to follow those contractions where you actually see the dynamic condensation of F actin. And it's very well polarized and it's propelling the nucleus forward. That's a representative control. For the mutant, what we see is that the distribution of the contraction is more cup-like shape, and as compared to the control that you can see moving at the same time, there's no jump. The cell is gliding, right? And this is measured here. So we have not only changes in the distribution, but as you can notice on this recording, more intensity, so more contraction. If you target TTLL1, you cannot rescue this defect. So it doesn't seem to involve the microtubule network. It seems to be really affecting the regulation of those contractions. And the pathway that controls this contraction is well known, and we tested different entry points, and I'm not showing everything for sake of time, but what we saw is that MLCK is actually an ideal target because it has a genome-encoded glutamate chain, and actually we discovered that it can coexist as long and short form. So if you lose CCP1, what you have is an unbalanced proportion, so you are accumulating those long chain, MLCK long chain, and this has an impact on the contraction. Why? Because we discovered that the long chain is not more stable than the short chain, but is actually more efficient, more active. And the readout, so you can see that here, and the readout is on the target. You can see more phosphorylation of the light chain, which then allow more activity of the myosin 2 complex. And this is how we believe losing CCP1 interfere with the migration of those interneurons. I would say that the most striking defect that we observed was indeed when we were looking at the front of migration of the whole population. So you are at E13.5, you see the cells that are invading the cortex. We had supernumerary interneurons, and this is the first time that I see such a defect. And this is true at all rostrocaudal region that we analyzed. If you now look at the apico-basal distribution of those neurons, you are basically binning the cortex, right? Black is control, red is mutant. You can, if you focus on the black, you can see actually the two streams. So more cells are here and are there, superficial stream, intermediate stream. We have only a defect in the intermediate stream. The reason is not clear. Could be that these guys are more free to move as compared to those ones that are a bit more squished under the pia. Could also be cell intrinsic differences, because as I told you, the ones that are migrating there are mostly somatostatin positive. So we don't know exactly, but at least what I can say is that we have more of the supernumerary interneurons located in this intermediate stream. And this is true at birth, but this phenotype disappears at P8. And if you look at adolescence, if you look specifically at two broad subclasses of interneurons, the PV for paravalbumin or somatostatin interneurons, neither the number nor the laminar distribution is affected. So this is only a transient defect. How is loss of CCP1 transiently increasing the interneuron sorting in the cortex? That's a tough question. And we spent quite some time to try to understand this. I told you that the average speed of migration is not different. The regions that are producing those interneurons are also feeding to some extent other regions in the forebrain, septum, striatum, olfactory bulb, and so on. So we looked for phase change or rerouting, and that was not the case. There's no non-cell autonomous defect that would arise from CCP1 knockout cells on their own progenitors for expanding their pool. No. The survival doesn't seem to be affected overall. So how come then you're not producing more cells? You're not having more survival? How come you get more cells at certain time point in the lateral cortex aggregating in the knockout? And we came with this provocative idea that maybe changing the behavior of these interneurons at the cell level would have an impact on the population of interneurons. The reason is that we know that these interneurons are not migrating in a synchronous way. If you remember the movie that I show you, one cell is stopping, the other one is moving, the other one is jumping. So there's no synchronicity. So what we tested was whether losing CCP1 inducing the gliding would change the synchronicity of movement at the population level. That's also a tough question. And for this, we had to ask help from Mohit Adhikari, a physicist, to model the migration. And for this, he used actual recording for our time lapse. And he did some different type of analysis. And I'm just showing one here, which is pairwise analysis. So you would pick randomly pairs of interneurons to see if they are moving together, right? Wild type or conditional knockout. And he saw that they were more moving together, suggesting that there's more homogeneity of movement at the population level when the internals are not expressing CCP1. And I asked him, wait, can you raise these cells? He told me it's impossible. You know, on your recordings, they are all starting from different points. So what we did was to create surrogates, basically taking specific information such as the instantaneous speed of the nucleus, the frequency of nucleokinase, and so on and so forth for both categories. We shuffled information, and Mohit created surrogate neurons and erased them. And this is the 100 wild type or CCP1 surrogates. You can see this bar, which represent 80% of surrogates ahead, and you see the advantage at the population level. And this is true for some distance from the start, which is in agreement with the observation that we had, because we see mostly the defects in the lateral dorsal cortex to some extent, but it's really fainting in the medial cortex. Can we look back this to experimental biology? So for this, we use microfluidic devices where we seeded some cells and we looked at the movement in grooves. Maybe you can see it, I hope. So the grooves are basically coated with laminin and fibronectin, and you see a single cell moving. What you can notice here is that the cell is undergoing a saltatory migration, which means that this behavior is at least partly cell intrinsically encoded, which makes sense with CCP1, right? And if you lose CCP1, this one is gliding with our neighbors. And it's not expressing CCP1, really suggesting that CCP1 belongs to a cell intrinsic machinery that controls the saltatory migration of those interneurons. You can even raise these cells. So if you plate together control that are yellow with conditional knockout that are green, after a while, you can see that the green are winning. So if you measure the speed of migration, there's no differences. Again, the total time of nuclear posing is reduced, as it is the case in situ, in the brain slice. And of course, there's also a reduction in the instantaneous speed of the nucleus because there's no acceleration that are coming with the jump, right? And again, you can raise these cells, and you see, again, an advantage at the population level, which is basically taking up the model from Moit. There's another interesting, so basically this is how we explain, this supernumerary internals that are reaching the cortex, mostly in the lateral cortex, right? But there's another very interesting observation. I mentioned that these cells, the supernumerary one, are mostly enriched in this intermediate stream, right? And this is a region where you've got lots of progenitors, including these transient amplifying progenitors, that generate projection neurons. We are at E14.5. This is a time when these cells generate superficial layer neurons. And if you look by super resolution microscopy, you can see that, so we are looking here, this interneuron, for instance, is in close opposition with intermediate progenitors. And these guys have been labeled through electroporation of a membrane cherry under a regulatory sequence of TBR2. So this suggests that with this close opposition, maybe these guys can talk to each other. Is there any crosstalk that could happen there? And this is actually the case. So when you look at the proliferation rate, it's actually increased in the conditional knockout for only these progenitors, not the apical progenitors that are not flooded by the supernumerary interneurons. So this suggests that there's a mechanism that allows these intermediate progenitors to read the number of internals that are migrating. If that's true, if you have less interneurons, you would expect the opposite effect. And to test this hypothesis, we had a nice collaboration with Nicoletta Kesaris in London, who provided samples from the NKX 2.1 knockout, where there's no interneurons migrating at that stage, and where we saw a massive reduction of proliferation only of the intermediate progenitors. So what is the long-term impact of being overflooded with interneurons? Well, we saw an increased number of superficial projection neurons upon loss of CCP1, specifically in these migrating interneurons. Maybe there's a defect in the balance of excitation. This is something that we are looking at actively. What is the nature of the crosstalk? What we can tell you now is that part of it is diffusible for sure. So if you culture those intermediate progenitors on top of millicells, with or without interneurons underneath, you see that when you have interneurons, there's a boost of proliferation, whatever the genotype is, meaning that this is not because the interneurons are not expressing CCP1, that they are boosting the proliferation, but it's really dependent on the number of interneurons that are close to those intermediate progenitors. We're looking actively now at the nature of the crosstalk. We have several ideas about it that we are testing in the lab. For this part, take-home message. The first is that we discovered a new biochemistry way to control MLCK and so the actomizing contraction because CCP1 is involved in the digestion of the genome-encoded glutamate chain. Now we understand the posing during migration. This is allowing to provide some asynchronicity of migration for the population, which is important for proper sorting of those interneurons. And we discovered that this sorting is important because there's actually a crosstalk between the interneurons that are migrating and the intermediate progenitors. The nature of that crosstalk is still unknown. What I can tell you is that it's a true crosstalk because we have the opposite effect from the intermediate progenitors. And this is a nice paper published by the Broccoli lab a couple of years ago, showing that some chemokines are actually involved in the attraction of those cortical interneurons by the IPs. So we have here a mouse model. They are also alive during adulthood. We have a mouse model where we would like now to, well, we have expansion of superficial layers. And the question now is, what is the impact on the behavior? And so we basically analyze these mouse using a behavioral screen. It's a collaboration with Olga Peña-Garicano in Pays Basque in Spain. And we noticed that there's actually no major defect in olfaction, anxiety, there's no stereotypy, recognition memory is okay, and there's a slight increase of activity. Locomotion is increasing in the open field test, but not so much, I should say. What is very interesting is that when you look at separation calls, so just here, you can hear them because we transform those 20K hertz call. It sounds a bit like a bird, sorry. But basically, the idea is that you separate the pups from the mother, and the pups will start emitting those ultrasonic vocalization. And what we found is that pups from the conditional knockout were actually providing more and longer duration calls. And this is interesting because this has been shown in several models for ASD, autism spectrum disorder, including the famous Fragile X model and the spontaneous BTBR model. Well, not saying that these animals suffer from ASD, but this is one ASD trait, I would say. So then we looked at sociability because sociability is often impacted by ASD. And we did this 3-chamber assay for social preference. So you have the congeny here. The mouse will spend a lot of time sniffing around, being interested by the new congenier, and there's no differences between genotypes. What we found is that if you place a new mouse, the interest now is driven toward this new mouse in the control, but there's actually no interest per se by the CCP1 conditional knockout adult mouse. So there's a specific deficit in social recognition that is quite interesting and that we would like to better understand at the connectivity level. And that's something which is done by Romain with a PhD student supervised by Carla in the lab. So also another observation which is interesting is coming from clinic or clinical studies that are showing that a lot of patients suffering from ASD show actually too many numbers in specific regions, such as the prefrontal cortex. And brain imaging study also show increase in thickness, suggesting that this is maybe something which makes sense for our mouse to look at. And there's also this beautiful experimental paper from Nancy Hip lab, which shows that if you increase the number of upper layer neurons by playing with wind signaling on the IPs, you actually get animals with more superficial layers, neurons, that show ASD-like traits. So what we're doing now is try to refine the place in the cortex where we have this expansion of neuronal numbers. And what I can tell you is that it's very anterior. And you can see here the motor cortex. And like I said, this is a work done by Romain. Romain is also actively tracing the projection because we want to see if there's any change in connectivity, which is making sense also, again, in the context of ASD. And in a more unbiased way, a more global way, we're looking at the connectivity by DTI or resting state fMRI to look at long-range connectivity defects possible in collaboration with the GIGA Cyclotron Center that we have in Liège. And this is where we are with this project. ASD kind of makes sense as well because we know that some patients suffering from mutation in CCP have behavioral defect that looks like ASD. And so we had a chance to get a hand on fibroblast samples from these patients that we have been reprogramming. And what we're doing now together with Bernard and Tonella, and also under the guidance of IRA, we are making assemblies to look at the migration of interneurons, but also at the growth of axonal connection between different bits of 3D structure. Let me now go to the second part. And this is really the new part, which is not published. It's about
Laurent?
OPC. Yep.
Just for your information, it's 6 now. So how much more time do you think you have, more or less?
I think I have, if I can have like 5-6 more minutes, because this part is sure. Okay.
Yeah, just to give you feedback.
Thank you. So these oligodendroglial progenitor cells are actually born in different ways in the forebrain. You have 3 ways. You have the one that are born during brain development, and that we are interested in, of course, and the one after birth, which is born from dorsal progenitors. This third wave is actually the one mostly involved in myelination, together with the second wave OPCs. We do not know much about the first wave, sorry. And so we are interested by those cells to see whether they have a function during cortical morphogenesis. So what we know about these waves is that they were firstly described by Nicoletta as originating from the MGE and the AOP. And Nicoletta, at that time, with the tool of that time, concluded that they might not be involved in myelination as they die after birth, I think around P10. But very recent work from Gonzalo Castelo-Branco actually showed that by single-cell biology, these guys are still present after birth, they are just outnumbered. And now you have also this functional analysis done by the Cecilia Angulo lab together with Alessandra Pirani, that show that these OPCs can form GABAergic synapses with cortical interneurons that have a shared embryonic lineage. And this is peaking at P10. And these guys later on develop myelin and are involved actually in myelination of interneurons and also to other neighboring neurons that are part of the same network. So what we decided to do together with Carla, who now supervised Fanny, a PhD student, is to look at the migration of these cells because not much is known about it. And they compare the migration of cortical interneurons with the OPCs. So the interneurons are using 2 streams, like I mentioned. The OPCs looks like having a different path behavior. They use more like a random walk, at least at the early step of corticogenesis. If you compare the parameters of migration, the most striking differences is about the amplitude of nucleokinesis, but also the total time spent by the nucleus posing. It reminds us somehow the CCP1 conditional knockout. And so using the web tool from Gonzalo, we looked at the expression of CCP1 in these cells, and it's actually not found, suggesting that maybe, and it's purely speculative here, CCP1 is also part of the machinery and it makes the difference, I would say, between the behavior of those OPCs and interneurons, at least at the onset of corticogenesis. Later on, these cells, the interneurons, are undergoing this intracortical dispersion, and you can see almost a mutually exclusive distribution of the OPCs that are a bit kind of stuck on the region populated by fibers, and they enter in the cortical plate using a radial path. So we looked whether these OPCs may use fibers from apical progenitors, and actually that's not the case. If you look here in white, these are vessels. You see that these OPCs are actually migrating on top of vessels, which is in accord with work previously found, published, on OPCs that really like to move on vessels. So again, now if you look at the parameters migration, they are totally different. The reason is that you got more and more vessels. You have a progressive angiogenesis that starts in the ventral part and continues in the dorsal. And when you look at T16.5, the OPCs are mostly sticking to the vessels. So to try to understand the function of those OPCs during cortical morphogenesis, we use this combination transgenic combination to specifically eliminate those OPCs using expression of diphtheria toxin at 11.5 and 12.5. And I should say that we checked a lot of parameters, no difference in survival or proliferation or differentiation in most cells of the forebrain. The striking defect was the migration of the internals. It's really strong. You have a major defect in migration, which is actually increasing with time, which is also correlate with the increased growth of the blood vessels, right? If you remove the OPCs, you see different behavior. So while the internals are freely moving in the cortical anlage, they seem to cluster around the vessels. And this is shown here. The distance between the neuron and the vessel is almost not moving when you have no OPCs around. So we believe that without OPCs, the internals are kind of sticking a bit more to the vessels, which is not natural for them. What is the mechanism? So we're looking at that. We are culturing here POA explant for OPCs, NG explants for interneurons, and we looked at the migration and the mixing of these cells, and they're actually not mixing. You can see that they are touching and kind of repulsing themselves, and this guy is kind of playing ping pong with two OPCs. So there's a signaling pathway involved in repulsion that we're looking at right now. The working model, because it's still a working model, when we see that both the interneurons and OPCs are migrating at the same time during brain development using different paths, we see that the OPCs likes to stick on vessels, and to some extent, we believe, prevent the internals to stick to the vessels. When you lose OPCs, and that's of course shown here, you see the OPCs on the vessels and the interneurons out. And when you have no OPCs, well, you have a defect in migration, mostly due, we believe, to the defect in migration due to the attraction to the vessels, right? Mechanism is on this way. We are actually validating a lot of pathways right now. And this is my last slide. So I'd like to sum up what I just mentioned. So today I wanted to show you how migrating cells during development engage into crosstalk with their environment to shape cortical morphogenesis. And for the first part, what we showed is that the internals enter into a dialogue with the intermediate progenitors to control the output of neurons. And we know that this is bidirectional. We don't know what is the nature of the molecule diffused by the cortical internals that have an impact on the intermediate progenitors. If you mess up with this regulation, you can end up with behavioral defects, some suggesting is the behavioral traits. For the second part, I showed you that the OPCs seems to allow the internals to move freely by preventing them to adhere on vessels. And here there's a huge complexity in terms of network and in terms of crosstalk. Part of it is already known. I'm not going into much detail, but we're still looking at those question marks here. So I'd like to, and they likely interplay between, of course, those two systems. So I'd like to end by first thanking the people who were directly involved in these stories. It's about actually 8 years, and it's to be continuous. So it's all started with this trio, Juliette, Sophie, and Ariel, who were interested by the basic mechanisms of migration in health for internals that allow us later on to better understand what happened in disease. And this is the work from Elise and Carla, which is now also including Romain. And this opened door for looking at OPCs with Fanny, also supervised by Carla. Carla is really a central person, and she's available and on the market looking for a PI position. And we are also now starting to model using IPS these crosstalks, thanks to the help of Antonella, Bernard, and Ira, who just joined the lab. And also, I'd like to thank all people from the lab for the feedback that they gave on those different stories. We have actually different topics. I had no time to show everything. I'd like to thank the collaborators. I'd like to thank the funding support, and I'm now ready to take a question that you may have. Thank you very much for your attention.
Thank you very much, Laurent. I hear an echo there. I don't know where it comes from.
Yeah, I can. Yeah. Thank you. Sorry, I've been a bit long. Sorry about that.
It's okay. It's okay. People stuck around.
Cool.
Thanks for sharing this wonderful work, driving us through the cortex from excitatory to inhibitory, from ventral to dorsal, and these cell-cell interaction, non-cell autonomous aspects were really exciting. So you're back on the regular window. You're seeing everything.
Mm-hmm.
Yeah.
I see everybody. Thank you guys for being there.
So a few questions we do have time for a few questions.
Okay.
And I'll take them in the order you guys voted for them. Do you think the gliding phenotype is only actomyosin dependent? Is there any involvement of microtubules? Maybe short answers if you can.
So I guess that's related to the first part, right? Where we saw the importance of this actomyosin contraction for, it's not really in terms of guidance, right? We're talking here about getting more neurons at a specific time. So we're changing the way the engine is working and so the behavior of the cells. I don't think we are really dealing with guidance of interneurons here.
Okay. Probably with regard to the second or the middle part, are the extra upper layer neurons only present in the lateral cortex where you see the supplementary, the supranumerary interneurons? Is the number then adjusted postnatally if the final number of interneurons is not changed? That's from Isabel. Hi, Isabel.
Right. So there's actually a gradient of defect which match the gradient of migration defect. It's mostly on the lateral cortex and in the more rostral part where actually we see the most defects in migration. The superneural neurons are still seen after birth until at least P21. And we have defects in the adult behavior. So we believe that they are still there.
So I missed the beginning of your answer. You're saying it's more in the lateral part where there are the...
So I said that it's actually following the gradient of defect that we see for migration, which is more lateral and more rostral.
Okay. Any difference in the way MGE and CGA-derived interneurons migrate?
Yeah, it's a good point. We've been looking, unfortunately, only at the DLX driver, which cannot help us to make a difference. I think that are cool things to do. What I can tell you is that the migration is changing also during time, and it's likely to be also specific for some subpopulation that may come from different places, but they are very difficult to identify because there's not much of markers that we can basically use. Okay.
Does any of the cytoskeleton regulators that you have described impair more than one? Oh, sorry, let me read it through first. Okay, yeah. So do any of the cytoskeleton regulators that you've described impair more one cellular process than the other? For example, migration versus branching. Related to that, are they interneuron specific or do they also function in glutamatergic neurons?
Yeah, that's a lot of questions. So obviously, when we at the different molecule that we looked at, the impact is different. Sometimes you got more defects at the nucleokinases or more defects in the branching. For instance, the glycine receptors defect seems to be more affecting the nucleus movement. And for specificity for projection neurons versus interneurons, keep in mind that the projection neurons are actually locomoting. They're undergoing some slight saltatory movement, but that have nothing to do with what we see in these interneurons. And what I can tell you, for instance, is that P27 mutation also affect the migration of the projection neurons, but not by affecting exactly the same process, I would say.
Okay. How do the migrating interneurons know where to stop? Are there phenotypes observed where they migrate longer?
No, it's a cool question. I think, so we have not looked at that, of course, but there are papers where, if I remember well, some guidance cues are also involved in stopping. I think I'm thinking about GABA, for instance, through GABA B receptors. So there are like cues that are informing these cells to basically stop at some point.
Okay. Role of the Golgi apparatus in the migration?
Where it is? Sorry.
Role of the Golgi apparatus.
Oh, yeah. Not, there must be a function, of course, but I'm not aware about a study that actually looked specifically at that. It must have a function, of course, and impacting with Golgi should impact a lot of things during migration. But yeah, I'm not aware about a specific work on this for interneurons.
Interesting question here. Do interneurons migrate in the adult brains? Meaning, I guess, is there any reason to think that they might be more plastic than the glutamatergic guys, and that maybe in case of lesion, they could migrate somewhere? And have you ever conditionally knocked out CCP1 in the mature brain?
Yeah, it's a good question. In the mouse, there's no more interneurons migrating in the adult brain, in the cortex. In the adult brain, actually, there's a lot migrating in the rostral migratory stream, where CCP1 may actually have a function as well, right? For the cortex, maybe upon lesion, you can have migration of some interneurons, but again, I'm not super aware about this thing. Mutating CCP1 in the mature brain, yeah, you know, I'm not sure that this will really help understanding that question. CCP1 is involved in the deglutamylation of microtubules, and you would affect a lot of microtubule-related processes such as axonal transport, which would impact the survival and the function of those neurons. So I don't think you can easily look at that question. I'm not sure that it's so easy.
Okay. Did you check whether knockdown of any other TTLL protein might rescue the CCP1 phenotype? Yeah,
I think we tested at least 2 of them, but again, what was rescued was the branching dynamics, but not the movement of the nucleus. And if you remember my presentation, the genome-encoded tail of MLCK cannot be reconstituted by TTLLs.
Okay. How do you know the increased proliferation in the cortex of the CCP1 knockout is not caused by defects in migration of oligos rather than CINs, I guess, cortical interneurons? Yeah. Yeah, I guess related, kind of fusing your two stories.
So two remarks about that. So we used a specific driver for interneurons. So to answer this question, well, so we're not supposed to impact I would say directly on the OPCs, even though fractalin, which is released by interneuron, may impact on the generation of OPCs. Huh? That's a work published by Anastasia Vorovona. Also, what we saw from the database is that CCP1 is not expressed in OPCs. So, okay. Yeah, that's what I can say. But one should look at it.
Okay. The usual question about circuits. Yeah. Are CCP1 knockout interneurons well integrated?
No, it's a good one, and this is something that we're looking at now. We are also, in that respect, planning to drive the recombination under GAT2-CRE mouse, ERT, to basically remove CCP1 in settled interneurons to make a difference between losing CCP1 in mature neurons and losing CCP1 during the development and the migration of those cells. But that's, of course, interesting question that we have to look at.
Okay. Considering the repulsion between interneurons and OPCs, do you have any thoughts on radial dispersion of interneurons and the change in the distribution of OPCs from early to late embryogenesis, basically the developmental time course of this?
So we believe that, so this is also related to the vessels that are growing from ventral to dorsal forebrain. And so the interneurons are actually migrating from the ventral place, like the OPCs. And at the stage when they are migrating, the OPCs are already there and covering the vessels there. But if you look in the cortex, the OPCs doesn't seem to migrate on vessels because there's not much vessels, right? And they move randomly. And we believe, and we are actually doing some recording using 2 colors now, that there's really a repulsion occurring at all stages of development. But the nature of the signaling, we're looking at it, of course.
Okay. Do you think there's a relationship between behavioral social differences in the CCP1 knockout and parvalbumin interneuron dysfunction in ASD and schizophrenia?
Yeah, it could be. I mean, this is a very specific, social recognition is very specific. So there's no big other social defect that we saw. What we think it might be linked to the network, specific network. There's one between the Cortex and the VTA, for instance. So we should spend some time looking at that. Yeah. But yeah, I cannot answer for sure.
Okay. A question from Nicoletta: what happened to the overall distribution of interneurons? Oops, it's moving. Voilà. What happened to the overall distribution of interneurons within the cortex when OPCs were eliminated, apart from sticking to the blood vessels?
Yeah, that's a good one. The problem that we face is that the embryos are dying. So we can push till E17.5, E18, I think. So we cannot really address that question. We need to find another way to do it. But that, of course, would be very interesting to see. If you are changing the way these interneurons are migrating, will they reach the right position? That's a good point. Yeah.
Okay, 2 or 3 more questions. Since the supranumerary interneuron phenotype in CCP1 mice is transient, is the non-autonomous impact on basal progenitors restricted to distinct temporal cohorts of excitatory neurons?
I think we showed that. Huh?
Oh. Say that again?
No, I think we just showed it. So yeah. It's actually the one that are born around E14.5 when they are flooded by those supranumerary interneurons, which are actually sitting in the upper layers mostly.
Yeah. And then a difference between lateral versus medial, you also gave us the answer. Yeah. Okay. How is CCP1 expression changing in later change stages? Does it get lost, or might it have additional functions aside of migration control?
Yeah, so we have never looked really in the lab. What we know is that CCP1 through polyglutamylation regulation is also important for trafficking of receptors that then will maybe change also the way synapses are working. But this is also something that we would address with the GAT2 CRT system.
Okay. And one last question, Laurent. Did you find any changes in the anterior commissure axonal projections of CCP1 conditional knockout? So I guess relating to the projections of these.
Yeah. So I cannot really answer right now because we are actually processing this information. I cannot tell you for sure that the days will not change. Sorry.
Oh, there's an important question from Alain. I think you might actually like that question.
Yeah. So I saw it. So I can show you, Alain.
What does the round sculpture behind you represent?
So it's basically a Modigliani hidden place, right? Through a piece of wood. Yeah, obviously I'm not in the lab.
Thanks to you, this show is going to be automatically censored now, Laurent.
All right. I had a great time. Thank you very much again for the invitation. That was cool.
All right. Well, thanks for being here. Thanks to everybody that attended. Next week, if I'm not mistaken, I think it's going to be Pierre van der Hagen. So we're staying in Belgium.
Oh, wow.
Same place, meaning in front of your computer. Same time, meaning Thursdays at 5 p.m. Until then, take care, and I hope you have a nice end of week and weekend. Bye-bye.
Bye-bye. Thank you all.